So hello everybody and welcome to another SciNote Webinars. We're really excited to bring you this one. It's actually the most what we expected to be the most attended webinar so far. It's titled How to Use an Electronic Lab Notebook to Scale Your Processes. I'm Klemen, the founder and CEO of SciNote, and I'm really excited about today's host, who is Rita. She's team leader in molecular biology at a company, Ingenza, from Edinburgh, Scotland, and she will explain more what the company does. But she has been using SciNote for 4 years, and she probably can teach us some things about SciNote that we didn't know. So we are really excited for her to share her knowledge, and hopefully you guys can learn, and I hope you'll find her tips and tricks and experiences from using SciNote helpful. So a couple of things before we jump into Rita's presentation, just some housekeeping. This webinar is being recorded, and if you have registered for this webinar, you will get the record of it, so no need to write notes. We'll begin with Rita's presentation, which should take about 25 minutes, and then we'll go into the discussion, and lastly we will answer as many Q&A, so questions from the Q&A section as possible. So with that, there is a Q&A section at the bottom of your screen. So at any point during this webinar, you can write the questions there, and as we go along, I will collect these questions, and I will ask Rita about what her answers are. If your question doesn't get answered, we will follow up with you after the webinar, so definitely please give us the questions. This webinar is for you to learn, so definitely feel invited to participate. Before we begin, last thing, we'll do a quick poll just so that we understand who are you guys. So what are your needs? At what stage you are in? So the first question is, how far are you in the transition process? So you'll get a couple of options that you can choose from. And while we are doing that, I would just like to do some product placement. Here in the bushes, we have our brand new book. So this book has been published by our team members together with Wiley, about the digital transformation of the laboratory, and it's full of stories similar to those that you'll hear today from our users, what their challenges were, how they overcame it. So yeah, if you're the reading type, definitely go look for this book. It's so new, it's still in the plastic foil, so yeah, be one of the first ones. All right, so I think we're done with the poll. So without further ado, hi Rita, and the stage is yours. Welcome to the SciNote webinar.
Hi Klemen, thank you very much for inviting me, and I look forward to share our experience here at Ingenza with SciNote and with this transition. I certainly remember it was a very stressful and difficult decision at the time, so hopefully today will help some of you that are tuning in. So let me just share my presentation. It's all okay? Yeah, full screen?
Yep.
Cool. So as Klemen said, my name is Rita. I'm a molecular biologist by training. I work at Ingenza, where I'm responsible for microbial strain engineering. To tell you a little bit about Ingenza, We are based just outside Edinburgh in Scotland. In the UK, we are an SME, a small medium enterprise. That means we're under 50 employees. And we are a contract research organization. So we get different customers that come to us with particular issues, and we specialize in the design and the optimization of biomanufacturing processes for chemicals, proteins, pharmaceuticals, and also biofuels. We are a very multidisciplinary team, and we've worked with customers and universities in several applications in a diverse range of projects. So we've worked with customers and universities to develop, for instance, biomanufacturing routes for petrochemical replacements. We've worked to try to identify new and novel antibiotics and how to biomanufacture them. We also worked with customers to improve food productivity in a sustainable way. We have several projects looking at improving the quality and also the equity of healthcare worldwide. Particularly, in the last couple of years, we've been looking at production of vaccine components to tackle the SARS-CoV-2 pandemic. And we also, as I mentioned before, have customers looking at sustainable liquid fuels. So we have not only a diverse team, but also a very diverse range of projects. Just so it makes a bit more sense for you how we look as a company internally, we have 3 departments: a molecular biology department, which I'm part of, a fermentation, and the chemistry department. And we work with different bacterial, yeast, and mammalian hosts for all these processes that we work on. And any typical project will look like a customer comes to us, we do some strain engineering in the molecular biology department, we pass on that new host to the fermentation department, which will look at developing a fermentation or bioprocess. And then the fermentation department will work closely with the chemistry department, where there's some analytical method development and also downstream processing and product isolation. So in every project, all 3 departments tend to collaborate. So 4 or even 5 years ago, we were desperate to find a solution to improve the way that we were collaborating between departments in these projects and recording and managing the data as the company grew. So I guess if you're attending this webinar, I don't really need to convince you, hopefully, of this, but just as a reminder why paper lab books are so bad. You probably know they are very slow and tedious to write up. This typically involves printing, cutting paper, taping paper onto paper, or a long time spent writing up. They're obviously non-searchable, at least not in a straightforward way. They're also very expensive and difficult to store. So here you have a photo of our actual cupboards that still hold our old paper lab books, and these had to be fireproof cupboards that were quite pricey. And obviously they also occupy a footprint in our office space, so we were paying rent to keep these archaic paper systems. Sometimes, depending on the colleague, they might also be a bit difficult to read, and obviously they're not really a tool that promotes collaboration or efficient management of a project, because only one person at a time can use and read and make use of them, basically. So this was very outdated. So for us, it wasn't really a question if we should move to an electronic system, but when and which one. This is obviously because electronic lab notebooks in general promote fast and efficient and controlled recording of data. Also, because the provider has usually done some work in advance to show that they're compliant to several systems. This is already done by the provider, and we don't need to convince or gather evidence to convince our customer that we use a system that has quality and compliance. Obviously, it also allows to upload and integrate all digital data, which is not possible with paper. With paper lab books, we had to reference a digital location for a certain set of data, digital data. That location could potentially change in the future, and that reference would become outdated. So the possibility to integrate digital data is also a huge advantage. And it also improves project management and collaboration. So here are some photos of colleagues of mine. So at Ingenza, we use SciNote on the lab bench directly, either through tablets. Here, in all honesty, we mostly use the tablet for consulting a protocol or checking some data, not really to input data live, because it's not really straightforward or not really easy to type in a tablet as it is in a laptop. So we also have laptops or desktops on the benches, and colleagues can input the data live as it's generated. So it was obvious that we wanted to change, and that would be a positive change. So how did we actually do it, and how did we select SciNote? So it was important for us to define a person that would have that would dedicate the time to do the market research at first. So that was in the past, back then, that was me. And I know that that could be very overwhelming. Even 4 or 5 years ago, there was so many providers of electronic notebooks, and I was quite shocked. I had no idea there were so many. I'd heard of a couple, and I'd used a different one in a different institution, but I wasn't ready to really, for the amount of different providers that there is out there. And maybe that list has even grown more over the years. So then I also asked people that I knew what they were using. And whenever I approached a particular provider, I would also ask them for a referral for a contact of a customer that they had, that I could independently email them and ask questions. So I could remove SciNote or whoever it was from the communication and honestly ask them for their honest opinion. So that was helpful as well. And then when I had that huge list in front of me, the only way I could cut down into a reasonable number of candidates was to establish what I call the non-negotiable variables. So for us, those were the fact that the system needed to be browser-based. So that for us was important because we have quite standard laptops in the office and in the lab. So we didn't want to have a very heavy software that would slow down our laptops. Some people have an old laptop still, so we didn't want to risk having a software that wouldn't be workable for some of our hardware. It was also important for us that the system would allow a local installation. I know these days cloud-based systems are very safe, and some people say they're even safer than local installations. I'm not an expert on that, but I know that for us, and to convince management, and so that we'd had a clear conscience of what we could say to our customers from a psychological point of view, it was important to say, look, we have complete control of your data, and it's in a local installation, not a cloud-based server. Because we're also involved in many projects that are run to GMP principles, it was important for us to have, for instance, FDA 21 CFR Part 11 compliance. And as for most of us, it was important that for it to be an affordable system. So that was also a big factor. And at the time, SciNote did have, it was more competitive price-wise compared to the other shortlisted systems. Still, other factors that then I took into account was whether the system would have a database function that would allow us to introduce, to upload databases that we use from, for instance, for our strains or our DNA constructs. It was also very important to show that we could export data easily. So that was something that people here were very concerned about. So what happens if one day, for some reason, SciNote disappears or the server breaks or something terrible? Do we lose all the data? So it was important to make sure that it was easy and straightforward to export whatever data we input. And SciNote has a reports function, which I quite like, which lets you tailor and choose what you export, either in a PDF or a Word format. And we use that regularly. Because I mentioned before, we have 3 departments that collaborate quite closely. However, they tend to have different, let's call them lab book pages. It was important to be able to internally cross-reference within the system to data from each department. So for instance, I might generate a new microbial strain in my SciNote page. I gave it to fermentation, and when they input the fermentation data, they can create a cross-reference to the page where I described how I genetically engineered that strain. So if there's any question, what gene is in this strain, or what antibiotic resistance, that might not make sense for all of you, but any detail that is needed, it's easy to click and go and check what someone else in a different department wrote. And finally, it was important to know what was the user perception. So this may be a little bit more, I don't know, less important for some people, but I wanted to make sure it wasn't a horrible system, because we had to use this every day, right? So it was important that people would turn it on and would have a positive reaction to it. And some of the systems I tried back then, some of them looked it didn't look appealing. So that factor also played a role. So after we shortlisted, at this point, I think I only had a couple of providers that I chose here. We set up a trial period with a trial team. And it was important for us that this trial team had a member from each of our departments. And we trialed the systems with dummy data. So in this trial period, we also had a close collaboration with the support team. And we would ask them regularly how to troubleshoot or how to do a particular task, or if we come into a problem, they would always help us sort it or suggest how to use the system, because we were still getting familiar with it. And for this, it was also important to define a deadline for this trial period rather than just leave it as an open-ended project. We told this team there's 3 months to do this, and at the end of these 3 months, we want to have the confidence that we have enough experience with the system that we can make a decision. And to motivate this team, the company also recognized their effort. So this was something that they could tell their line manager in their annual appraisal, look, I've contributed to improve the practices of the company by trialing these different ELNs. So this was something that also motivated people to do a good job on this trial and really do it thoroughly. Finally, we came after those 3 months, we did a group decision. And I think this was important, rather than have just just one person making the decision. It was important that people from the different departments could have a voice and say, I think this will work, or I don't think this will work. And actually what happened then, it was, yeah, this could work, but we still need to figure these things out. So that brings us to how we then implemented after making that decision. So mostly those things we had to figure out before implementation was to define the standard practices, or how we actually use SciNote. So if, I don't know, if maybe some of you will have already used SciNote or tried it out, and it's set up in a way that it has different levels. So there's the higher level is the team level, and then you can have within a team different folders, and then you get project, experiment, task, and then within the task you have different tabs as well, for instance, protocols and results. So there are different levels in the system, and those levels can be, and they are very useful to organize a project, particularly from a project management perspective. But when you're inputting data, sometimes two people that would input the same, an identical experiment in SciNote, they might choose a different way to do it, because it might make more sense for one person to use the protocols tab, another person to use the results tab, or some people break down every single tiny pipetting step to a task, and others will have a more broader scope definition for what a task is. So this would be problematic for us, because we need to make sure everyone does everything the same way, so that in the future we can all read and consult data that has been introduced by whoever, right? So we spent some time defining standardized practices. So the most common processes that we do in the company, whether it is fermentation or engineering a particular bug, we made sure we defined, okay, what is a task, what is an experiment, what's a project? And that would make more sense to you if you've used SciNote. So after we defined that, we also defined what we call the SciNote key users. We think this is very important to ensure successful implementation of the system. So these were three of our colleagues, again, one from each department. They are the people that are always here to hold anyone's hand through the system if they find it a bit overwhelming at first. And we chose these people. We thought it was important to choose these people, and they ended up being the same that did the trial of the system in the beginning. We thought it was important to choose people that had a positive attitude towards SciNote and towards change in general, because you don't really want to choose that person in the lab that is very stuck in their ways, and they think, why change something that it works? And I've worked with paper all my life, so I don't want to, this will just be extra work and create confusion. So you probably shouldn't choose that person as a key user, but you should choose a key user that has a positive attitude that will probably get that other person over the line and convince them this is actually a good change for everyone. These key users were then trained by the SciNote team. So they offer training in the beginning, and they also have lots of different training videos online and documents and things you can read. But they had like an intense training from the support team, and they learned about every possible thing you can do with SciNote. I have to say, until even having used SciNote for 4 years, there are more things that SciNote can do that we don't use yet. So I think it was important to make the decision that we didn't have to make use of every possible feature that SciNote has. But it was important to have the key users trained on every possible feature, so that one day if we decide to use it, they will know how to do it. Then those key users, they were the ones who trained all the staff. So we thought this was important, because having in-house training by your colleagues, it means that you're having training by someone who, you know, it's possibly your colleague and friend, you know this person, you're familiar, they speak of similar language than you do in terms of examples they give during the training, or they will use very specific examples to very specific projects, and they're familiar with those defined standardized practices that we defined in the beginning. So we thought it was important to have these key users do the in-house training rather than just sending everyone on the online resources that SciNote provides. And then we quickly realized that people tend to find the same issues and the same problems, and we try to continuously share tips and troubleshooting approaches. So I just put there 2 examples. So a very common thing that almost everyone complained in the beginning was, oh, SciNote doesn't do spell check. I thought that was funny, because when you write on paper, there's no spell check either. But thankfully, there is an add-on you can add to your browser. Maybe there's more than one. We use Grammarly, and that was a quick fix to have spell check as you type in on SciNote. Also, we realized after a few weird error messages that if you use symbols when you name files that you upload onto SciNote, that often gives an error. So we quickly shared that tip around as well, like don't use symbols when you name a file. And that is something that it's still ongoing, and also because SciNote are quite active in updating their system. Sometimes we need to share a new tip, or yeah, this is something that still happens even 4 years in, sharing tips and ways to make the best use of the system. So then I just want to give some example of how actually the current practices look like for us as a contract research organization. So whenever we get a new customer, we set up a new team on SciNote. Recently, they created a new level called folders. And if that existed in the beginning, I think a new customer would have been a new folder. So that's actually a good change, and we might consider changing the way we're doing things with this new level that appeared. But for now, when we get a new customer, we still set up a new team, a new SciNote team. And then the project manager for that new customer project goes in and adds the members to the team and defines and creates what's called on SciNote projects. And these are the equivalent to what would be like a work package in your project plan. And the project manager sets up these projects, assigns timelines and or deadlines and responsibilities and the users to each project. And then the what we call the technical lead, introduces templates for particular tasks that are, if that's relevant. So for instance, if there's going to be a fermentation at some point, we introduce this template. So this is a template that we can copy paste into different projects. Fermentation typically involves the same steps. So this was one of the standard practices that made sense to define. So for instance, they have media preparation, inoculum setup, sterility testing of the inoculum, set up the fermentation, and then at the end, results analysis, sterility testing. So this is a process that is quite defined, and they will use this template across different projects. So a comment here is one thing that we weren't looking when we were looking for an electronic lab book was a new way to improve project managing, and SciNote surprisingly was a very good way to became a very good way to manage our projects, exactly because the manager can create the different SciNote projects, assign users and deadlines, and the system reminds you when a particular task is due soon or if it's overdue. So these were features we weren't looking for at the time, but we were positively surprised, and now we've incorporated them in our day-to-day practices. So then finally, the members of the team will fill in the data in these prepared templates by the technical lead. So the technical lead here, by preparing this template, is really guiding you through, helping you prepare, set up your experiment. So that is very useful. And regularly, or when a project is finished, depending on the duration of the project manager can check the data. There are ways to mark each task as reviewed or done. So there are ways for the project manager to signal that they've checked this data, and then they can export all the data as a report. As I've mentioned before, this is a feature that we quite like from SciNote. And then we can export the data as either a Word or a PDF report, and we keep a copy of that in a different place that is SciNote independent. And that just gives us another extra level of peace of mind that if anything happens, we have that data in a different format somewhere. So The final topic I wanted to mention, how is this important to scaling up processes? So I mentioned in the beginning, we work in a diverse range of projects, and here at Ingenza, we work in a lab scale. So we have shake flasks at a milliliter scale, or fermentation vessels up to 20-liter scale. And in the past, we've developed several processes with different organisms that have been scaled up by our partners to 100 liters or multi-thousand liters processes. And this can be a very tricky process. And in order to ensure that this is a successful scale up, we need to ensure successful technology transfer. And key technology transfer tools include having a comprehensive documentation to guide the customer or the partner through this transfer. We need to be able to confirm regulatory compliance. We need to be able to provide full traceability in all steps of the process, the development of the process, or any manufacturing of small batches that we do in-house. And we need to be able to provide independent quality review and sign-off evidence. So these are all things that SciNote helps us do. And quickly, it became an integral part of our quality management system. It helps us to rapidly scale up with our partners processes that we've designed in-house. It provides full traceability of the development of these processes. We have peace of mind because we know everything is fully time-stamped, and there's an audit trail for the system, and we have daily backups of our data. And because we can export data from SciNote, we often will export SciNote data directly into either development plans or development reports, certificate of analysis, and often customer reports and presentations. So the work that you do in advance on SciNote often gets exported to important documents that we share with our customers as well. So yeah, this has been great and has worked really well for us. But of course, I'm only one person saying this. So I also asked my colleagues what did they think of this change and this transition. And I asked everyone if they want to go back to paper lab books, if they could. So overwhelmingly, almost everyone said no. There's always that one person or two that will always have the same opinion. It will be difficult to change their mind, but it's quite reassuring that no one regrets that change. I also asked them, how did they find the transition from paper to SciNote? About 40% said it was very easy. And over 50% said it was difficult at first, but now I find it easy to use. I would say that I would probably fit in this category as well, because it was quite overwhelming, particularly for me, because I wanted to make sure we made the right decision, and I was always trying to find issues with any system we tried. But now it comes quite naturally to everyone. And there are still the ones that still find it difficult. So you won't please everyone, but at least for us, the majority of our staff members are satisfied. Yeah, so I think that's all I had to share today. I feel like I've been talking for a long time. Sorry, Klemen. But yeah, we'll be happy to take any questions.
Yeah, thank you, Rita, so much for this wonderful presentation. Before we get into the discussion and all the comments, please, you are again invited to submit your questions to the Q&A section. You should find the two bubbles with the Q&A written below on the bottom of your screen. So feel free to write anything. We have a couple of questions already there that we'll answer shortly. And another thing that before we go into the discussion, we'll do the second poll, the last poll of today, titled, "What are your main struggles you're hoping to solve with ELN?" So while you answer this, I just want to mention just 3 comments that I have regarding the presentation. Things that I've heard many times before, and I would just like to emphasize that I think are quite universal, not just for SciNote, but any ELN. First one is the way you showed that you basically had to sit down and figure out the standardized practices and layers in SciNote. I think this is like really crucial because any system that you will get has some flexibility. So it's really important that decisions are made internally how to use the system. I think the second comment is that you said that you didn't go for all the features at the same time, or so a lot of customers that want to go, overnight from paper to notebook, which is it's a complex process, as you have seen. So definitely, baby steps approach is definitely the right one here. And third, that I think you did extremely well in your company, and it's I think often overlooked is the how you treated people in this process, like how you treated employees, like that you did recognition for their efforts, that you promoted key users here that were then becoming the internal trainers. So I think this is like really crucial that the team gets involved. It was a team decision about the system. So I think this is really crucial, key element here, because nobody likes to have their day-to-day operations kind of like pushed down from the management. So I think you did this extremely well. So I think, and the results from the survey speak for themselves. So good job, and yeah, great presentation.
Great.
All right, a couple of questions that I have straight from the bat. I found it interesting that you mentioned the cost of paper, because a comment that we often get is like, oh, but paper, paper is basically for free, and it's not. So it's no. Actually, when you start calculating and all the printouts and everything, not even taking the time into the account of how much time you spend this and searching, just like pure paper, it definitely does come to a cost. Did you do like any comparisons of like?
Yeah, at the time I did, because that was one of the comments, as you say, but this is so much cheaper than what you're proposing. And it is, if you just count how much we spent on each notebook we bought per year compared to our SciNote subscription, it's definitely less. But where people were then convinced is how much time this would save us. And actually, at the time, what I regret leaving out is the cost of the footprint that all the cupboards take up as well. And I think at the time I did count for the cost of the fireproof cupboards, because obviously, imagine if there was a fire for some reason, it would be quite, we didn't regularly digitalize every single page of every notebook continuously. It was something that happened every so often. But if there was a fire at any point, it would have been terrible. So the cupboards have to be expensive, fireproof cupboards. The footprint is massive. The time that you spend writing is massive. So I think it's difficult to put together all the variables to really do a true price or cost comparison. And I probably didn't do a good job at the time. But yeah, I think it's clear that it's a good choice.
And you touched on this a little bit from when you were talking about how you do work at the bench. Again, something that we get asked quite frequently, and it's not a straightforward solution, is if you want to, truly move to a paperless system, which, ultimately that should be the aim of everybody on this call. You should solve for, where do you put the data when you are at the bench? You mentioned you use tablets, you use laptops. What do you see like as the ideal solution for, hardware, what's the ideal hardware solution for end the bench? Or is it something more futuristic, like voice control or VR? What are your thoughts?
We had a lot of discussions about that because people were particularly concerned. Well, if I go away and do a quick measurement, for instance, I'll give an example that is common to us. If I do an OD measurement, like in a spectrophotometer, you take just a number. How do I make sure there's a piece of hardware next to that equipment and that equipment and that equipment, so I can always input? And then people were concerned that they would have to log in with their username each time they want to input some data in different parts of the lab. I think we worried a lot more about it than it actually ended up being a worry. And we ended up, obviously it still happens to most of us that you still have some piece of paper or notebook or an agenda or something that sometimes you scribble something on. That I can't say there is absolutely no paper used in the labs. That's not true and probably not realistic. So what we ended up doing is making sure that there was either a desktop or a laptop for a maximum of 3 users. So within 3 lab benches, there's a laptop that those people share. And it never really happens that you need to queue up for the laptop. That never really happens. And it ends up working well. And then there's the tablets that also about, I think that's less tablets, probably like 1 for every 5 people, I would say. And that still works as a way to like check what you've done previously or a protocol. As I said, you don't really type things in. What would be ideal? I don't know, some sort of robot that follows you around and reads your mind and types things as you think about them. I don't know, that would be the ideal.
Oh, no, don't spoil it.
Yeah, mind reading robots would be ideal. You imagine it and it appears. But I think having a laptop close by or it's good enough for us. And obviously then you might tidy up things as you process data at your desk back in the office. You also have access to SciNote, and then we'll tweak things or add things. So yeah, it works.
Okay. For you out there that are thinking about this, if tablets are your preferred way of going forward, we do have some partnerships with SciNote that can get you lab-grade tablets together with your subscription. So if this is your if you have interest in this, just make sure to write this and we'll get in touch with you. Okay, last question from me. What would you do differently if you went through the implementation again? Yeah, well, after actually watching one other webinar you had, what I would do differently is I would use the templates that I forget the name of the company that did another webinar with you, and they had an Excel template to score different ELN providers. From Germany, yeah. Yeah. So when I saw that, I was so annoyed that I didn't think of that at the time. And they have this brilliant scoring system that would have made things a lot easier. So I wish I would have used that Excel template. And I think it's available on the website and anyone can download it. Yeah, it was Norway Pharma who did it. And we will post the link to the chat, or you'll get the link to that Excel spreadsheet. Yeah, that's it was a very good way to tackle that huge list of providers. That it's really incredible, and I can only imagine that the list keeps growing. Yes. Yep. Yeah. All right. We received quite a few questions, so let's get started. So we have a question from SciNote Webinar. They are asking if we can give concrete examples of SciNote usage. So we actually have a web page of these examples, and you should be getting the link to this web page either in the chat right now, or we'll follow up with this link. So yeah, there are concrete examples, so hopefully that will be helpful. Then it's the question, Rita, for you. You showed the project outline, and the question is if that was the SciNote experiment. And
yeah, so it was a layout of different tasks inside an experiment. Yeah, so that's the experiment level, that's correct. And the tasks are the squares that you see. All right,
great. There is a question about the Lab Viva extension. So have you tried using it? It's the extension where you can order reagents straight from SciNote.
Oh, no, I'm not familiar with that, and we've not used that.
Okay. If you're having trouble with this extension, do reach out to us, and we can definitely schedule a demo with you to show you how that works. Then there are some very nice comments about the presentation being great. That's an interesting one. So the question is, does SciNote have a schedule system for equipment? So it doesn't have it right off the bat, but my question to you, Rita, is, do you manage equipment through SciNote? Do you schedule equipment through SciNote, or how did you organize?
No, actually, that would be a great feature to have. It's a mixture of two things. For some specialist equipment, we have a Google Calendar, and we use that. And other more simple equipments like incubators, for instance, we have a piece of paper that you can write on stuck to the equipment, and people will come in and book it for the next week or something. So having something integrated in SciNote would be great. Yeah.
We do get this question a lot. So without saying too much and getting yelled at by our product team, this feature should be coming in the future. So yes, it's definitely something we heard many times. Then it was a question about validation. So you mentioned that you have you work under the FDA compliance, the CFR 21 Part 11. You develop drugs, you do fermentation. So how do you make sure? I mean, SciNote is part of the QMS, as you mentioned, of your company. So how does the validation work?
Well, we got several documents from you showing that there was compliance to several different systems. So that is something that we just provide as evidence that we use the system that has electronic signatures, full audit trail. So it's, just by saying we use SciNote, that helped us, and because it's already validated by the provider, it helped us simplify that validation, if that makes sense, because it was done independently by a third party.
Yep, absolutely. And in our Platinum plan, we offer a lot of functionality that helps with validation. So specifically for customers that have high requirements for that. So we offer things like a validation instance, when you have an instance running in parallel for you to validate before the update is pushed to the production instance. We have scheduled updates, IQ, OQ, PQ documents. So there is a lot of helping materials if you want to run SciNote in a validated environment. Then it's an interesting question about animal cages control. I guess you don't work with animals, right, Rita?
No, we don't. Can't comment on that.
Okay, we do have customers that work with animals. There is no functionality that is dedicated to animal cages. However, we did have a webinar with Richmond University where they presented their workflow where they had animal cages. So expect a link in the comments with that webinar as well. So I hope you'll find that All right, another question. Okay, when I started working, when I started writing the experiment on the SciNote, it feels so much complicated compared to Perkinel and other ELNs I used.
Yeah, I think maybe they mean because you can't click a button and immediately start typing. You need to set up these different levels. And yeah, I agree that at first that sounds a bit overly complicated, especially when you see other systems. I don't think I'm familiar. Maybe I've seen the Perkinel, but I can't remember it right now. But I think over time, what you'll find is that initial setup is really worth it because it's a great way of organizing your project. So it may sound a bit overly complicated, but it's actually a great management tool, and it pays off in the end.
Yep. And I definitely second what you said, Rita. We heard this oftentimes that people just want to click a button and start writing. And we were many times tempted into reorganizing SciNote to be more. Yes, the thing is so SciNote was developed based on the research because people didn't really adopt ELN systems, or when they were adopted, they didn't really provide the values. Back in the 2014-15 when we did research, and what was missing was this some inherent structure that we found. What we have right now to be quite stable. With the additional layer that Rita you mentioned with the folders, we feel that this structure that SciNote sort of imposes on you is very sustainable and very universal. So yes, it takes some getting used to, absolutely, and in the future we'll try to add features that ease this transition, let's say. But the added value is that because you have a structure, a pre and post structure in place, everything down the line will fit into the structure, which will make your data much more accessible, findable. People will be able to understand it, once you leave or if you get a new coworker. So it's I would agree with Rita. I think it's well worth the time you need to put in initially.
Definitely.
Okay, another question. So I'm researching academic science in France. We already have a lab information system, lab collector, but no ELN. How did you implement ELN with existing laboratory information systems? How much effort did it take for the staff?
So we went through a bit of a different experience than this person, because we went from paper, from a very archaic system to SciNote. What we did, I forgot to mention that actually, is when we set up a date from which point we would use SciNote from, we only used SciNote for new projects that came up after that date. So projects that were already ongoing, we left them as they were with the paper system. We found again that keeping things as simple as possible promoted uptake and made the whole transition less scary. So it was really a case of starting fresh with a new project. So we never had to transition already existing databases or lab books into SciNote. Because a different customer is almost like a fresh start, right? So we did it that way, and it worked for us, if that makes sense.
Absolutely, and it's something that we recommend a lot. Because a lot of people, when they are looking to switch, they're thinking, oh, we have to do all of these projects now. We have ongoing projects, past projects, new projects. We have to transfer all to SciNote or any other system, which is a huge amount of work, and most of the time impossible, because it's not a way to import the legacy data. So yeah, we definitely recommend start with new projects and take it from there, and then you'll see maybe you don't even have the need to take the historic data into SciNote. Maybe it's fine where it is and just use it from now on. So it's definitely comes to the the baby steps approach we talked about before.
Yeah.
Okay, there is another comment about the rigidity of the structure of SciNote that we talked about. Did we ever discuss changing this or making it optional? I can say from our end that yes, we thought about this many times. Actually, so some that were SciNote users from the very beginning, we initially had only experiments and tasks. So the project level came, I think, like a few months in when it was too much of everything. So the one thing is nomenclature. So like maybe something that I would call a project, somebody else would call an experiment or even a protocol. So it really depends on the field. It does take some getting used to. We were thinking about having the custom names for these levels. In the meantime, however, we have seen this sort of nomenclature like project, experiment, task, or even a node to kind of starting to standardize in the industry. So it's a great question. I'm not sure where the future will take us. If you have any concrete suggestions, we'd definitely be excited to hear about it. But for the time being, the structure is here to stay. I don't know, Rita, do you have any recommendations how you would do it?
The recent change with the folders was really great for us, and that simplified a lot of things without getting into much boring detail. But often in a project, you'll have like work package 1 and work package 2, right? But then within that work package 1, you have 1.1, and sometimes we have 1.1.1.1.1, you know? So and it got a bit tricky to organize that with just the past three levels of SciNote, so the project, experiment, task. With the unlimited levels of folders now, we can compartmentalize and have an unlimited numbers of 1.1.1.1.1.
And you know, if you end up you know, running projects as experiments, that's totally fine. Like yes, it's a bit awkward because you call the same thing differently, but we have customers doing that, and it works. Although you know, I can see how it's maybe frustrating. Great question. Did you encounter resistance from the researchers? Like you showed, like there were a couple of those, but yeah, how was that?
Yeah, a lot of resistance in the beginning, and I think that's maybe natural. One thing that I always find funny is on SciNote, if you want to add a picture onto when you're typing in a text box, you cannot just drag the file, or yeah, you can't copy paste the file. So if you have a file, you can't do Control-C and then Control-V, and the image appears in the text box. And that is surprisingly very annoying to many people, which I found hilarious, because in the past that would have involved printing that image, going to the printer, picking up the paper, cutting the image, pasting it with tape, which you think it's a lot more than a lot more steps than just saving it as a file and then you upload as a file the image. It's really not that bad, but people will still complain about that. And I think we have like an annual catch up with the support team, and I think I have to mention that every year, because they ask us like, what would you like to see improved on SciNote, or have you had any issues? And I think I still mention the copy paste of image. It's fine, like it doesn't bother me, but it still bothers people. So people will find, because no one really likes change, so they will find the smallest thing. And I think the point that I tried to make, that you have the key users that have a positive attitude and patience as well to handhold and say it's okay, just do it this way, or I find it works that well this way and not that way. It helps. But yeah, there will always be resistance.
Yeah. Do you have any good tips? There's another question regarding that. Do you have any good tips on how, how to deal with those few?
Yeah, I think that having those positive people guiding the implementation in the company, like having really a chat with the key users and say, okay, we're tasked with this and we need to make sure it goes well, and it's actually our responsibility that it goes well. So you need to make sure you help your colleagues enough that it will be easy for them as well. So that it goes back to rewarding the staff members that take on that task as well. If you don't work in a team and you're just by yourself, I think it will still take some getting used to. And I think you need to accept that the first times you use it, it might not be the right way or the optimal way for you. And the next time you use it, you might use it a bit differently, but that's okay. And in the end of the day, it'll be okay.
as with everything, right?
Exactly.
Right, we are over the hour, so maybe just one last question, and then we didn't answer. We won't answer a few of them, but you'll get your answers in a few days. So, okay, last question: how did you organize user training by key users? Did they help trainings to their teams, or was there a few users who kept trainings to all end users?
I forget the options there, but the way we did it was we had the key users from each department. So the key user from each department trained their department, and they gave a few options of timings available at the time, because it's always hard to get everyone at the same time. So they probably had to do maybe 2 sessions per department. And every time we have a new staff member, one of the key users trains that person. It tends not to be department specific anymore, to be honest. Now it's whoever has more time available to do the training. But in the beginning, because it was a big transition, we made it more department specific, exactly because that person had the answer. So like, also when I do a fermentation, which level do I put the inoculum preparation? So the fermentation scientist could answer that specific question. Now we're all a bit more familiar with every department's practices, so the training is a little bit less specific. Who gives this training is more flexible, if that makes sense. Yeah.
All right, so we'll slowly wrap it up. Before we go, I would like to invite you for our next and last webinar of the year, which is a hot topic, as we all know. It's about security and scalability guidelines. We'll talk about cybersecurity, where is your data and how it resides within the ELN, and what are other quality assurance services that ELNs can provide. And with that, I would like to thank everybody. So all the attendees, all of you who were posting questions, much appreciated. And the most and biggest thanks to Rita. Thank you so much for being part of this. I learned a lot of new things. So
okay, good.
Personal thank you as well. And just based on the comments that we got, it seems you guys also really enjoyed it and found it useful. So thank you again so much, and yeah,
you're welcome.
Good luck with your company.
Yeah, good luck for you guys as well. We'll stay in touch.
All right, thank you, and thank you everybody, and have a good evening.
Have a good evening.